Okay, we've heard a lot about an exotic pathogen having a severe impact on native flora. I'm going to talk about an exotic host where a native pathogen is having a severe impact on it. I believe it's actually relevant to the considerations of the DIVAC working group. So I'm talking about Norfolk Island pines. Come on, zoom. Try the old-fashioned way. Okay, Araucaria is a genus of the Pinales. It's naturally restricted to the southern Pacific rim. Araucaria heterophylla, the Norfolk Island pines, are a very aesthetically attractive tree. They're tall, they're symmetric, they're evergreens. They've become a very popular planting in the Australian coastal areas for over 100 years. People have become very fond of them. They've become iconic. Of the 20 described species, some are endemic in eastern Australia, have you seen? The hoop pine, Cunninghamian, and bidwillie, the bunya pine, and more recently we've had the Wallamai pine described. The two fast-growing species have become popular in public landscapes because they grow quite rapidly. Heterophylla, the Norfolk Island pine, and columnaris, the cook pine, which is native to New Caledonia, predominate our coastal plantings, with occasional bunya pines and hoop pines in the Perth area. Norfolk Island pines have certainly come to predominate our Perth coastal strip, and in the last 3 years they have started into severe decline. We were first engaged in this. I run a commercial company that does plant pathology. We're often engaged by local government and by arborists to figure out what's going wrong with our trees. So we've been in the thick of it for a bit of a while. The question is, why are these trees declining? I start off with Norfolk Island, the natural habitat where these evolved. Norfolk Island's a very recent island. It erupted as a volcano about 3 million years ago. So it's 1,000th the life of the Australian mainland. The local geology of Norfolk Island is basaltic volcano. Basalt tends to weather very quickly, and it tends to weather into a pH neutral, very mineral rich soil. The soils on Norfolk Island are prone to mass movement of soil creep. We've been trying to unravel the genetic provenance of the diversity of Norfolk Island pines in Perth. This is an undocumented history. We heard before about the movement of different species into different areas. Because Norfolk Island pines have been here about 120, 130 years, we really don't know much about their provenance nor their genetic diversity. What we've been able to determine is their closest relative is columnaris, which suggested that seeds drifted onto Norfolk Island sometime after the island formed. The form of the tree has captured attention both because it's attractive and because it's very straight and strong and fast growing. And it was the British Navy who first looked into it, thinking it would make very good masts and spars. Unfortunately, the nature of that reaction wood is it didn't stand up to that maritime environment, and the wood largely failed. Why is unobscure, but plantations were started in Queensland, and they established that the life expectancy of a Norfolk Island pine is about 130 years. If we look at the climate of Norfolk Island pine, it makes you want to go there. The highest temperature they ever get is 25 degrees, and the lowest maximum temperature is around 19 degrees. The lowest minimum temperature is around 14 degrees. Also, the rainfall in Norfolk Island is very friendly indeed. It's between 1,300 and 1,400 millimetres. Every month gets good, steady rainfall. Compare that to the Perth area, where we have a drought condition in summer. We have very high temperatures and our maximum temperatures. We don't have that maritime influence that moderates temperatures. So our maximum temperatures are much higher, our minimum temperatures are much lower. Perth is a much more difficult environment. If we look at soil types in Perth, they're certainly not basalt. We are on the oldest sedimentary basin on the planet, and many of our soils against the coast are either the Quindalup dunes, which are marine, or Tamala limestone formations, and the pH is mostly alkaline. And the water holding capacity of our dunes is much lower, and the mineral nutrition is much lower. Nature of the tree decline is quite complex. There are most, if not all, coastal trees seem to have lost a lot of their foliage. Mostly, but not always, it starts at the top of the tree. Whereas trees further from the coast seem to be in less decline. Further, we find trees in severe decline adjacent to trees that are actually quite healthy. But this year, many of the trees have developed a vast number of distal cones. Cockatoos love this, but unfortunately the cones are so heavy that you get failure of these lateral branches and they break, and that creates an infection site, potentially. The leaves we find that have declined, many of them have black conidia. Mostly they're at the end of the growing tips, and that's very Botryosphaera-like symptoms of a fungal disease. We've noticed that many of the younger trees that have been planted seem to have a different type of leaf decline, more typical of Diplodia and Colicotrichum. The first trees we came across were at Esperance. This, unfortunately, is a slide that I sent some time ago. I checked it up. It was 2017. These were sent to Perth of trees that had a severe decline on the top of the tree. The bark had gone black. We isolated from that Neophusococcum parvum, which has been documented as a pathogen of Norfolk Island pines in Queensland. Only the tree tops were infected. We recommended that the top of the tree be cut off about ½ a metre below the infection front, and this had quite a good impact on the trees in Esperance. Many of them recovered and grew back, some of them didn't. In 2018 and 19, other large trees with quite different symptoms started to decline in Fremantle. They were considered, because they're in a populated area, to be a substantial risk. So those trees were removed. We took the opportunity to examine the trunk tissue from those trees and found that firstly, black stains had gone into the wood, and the vascular cambrium around that tissue had largely rotted. We isolated a different Neophusococcum from those trees, and that was Neophusococcum australe, which is known and identified in Western Australia as being hosted by tuart trees. And more recently, we've recovered the other Neophusococcum parvum from trees in spearwood. Now, these trees in spearwood are showing quite different symptoms to the trees that we first isolated from Esperance. And we've been identifying the DNA sequence in these fungus by using fungal intergenic spacer analysis to come up with a good confirmation that these are what we've seen. Neophusococcum australe in Fremantle, and more recently parvum in spearwood. The problem with Neophusococcums has come, with Norfolk Island pines has become much more prominent in the last 2 summers. I believe that I can now see impacts on virtually all of the trees in the coastal strip, but also we're seeing it in trees adjacent to the Swan River, especially in places like the South Perth foreshore and around the Peppermint Grove areas, Mosman Park. Trees further inland tend to be, but not always, in better condition. If you go down Lake Munga Drive, you will see Araucarias in perfectly good condition. They're robust and they're healthy. We note that trees, especially in the Fremantle area where there's roads and compaction, seem to have a much worse state of decline than trees that are in open areas, and trees that are irrigated tend to be much better. We've also noted that trees with the classic triangular form of a Norfolk Island pine seem to be worse impacted than trees with slightly different forms. Some are more columnar, some are very exaggerated triangles. These don't seem to be as badly infected. And that raises the prospect of, is there a genetic susceptibility to these trees to the state of decline? We've collected leaf tissue and soils from trees that are in good condition and trees that are in bad condition from a number of different areas in Perth. We've isolated more Neophysococcum that unfortunately isn't on the NCBI databases, so we don't know what it is. Getting sequence data of these plants is of these fungi is rather difficult because we're trying to do this fairly rapidly, and most typically you need to optimize the Petri dish conditions to isolate an extended culture so that when you go to the expense of sequencing it, you know what you're sequencing. So we're moving to different sequencing methods now. Most recently, we found something that is really quite extraordinary. We have in other species found gall-forming insects, and we were quite surprised to find on these trees, and Curran over there was the discoverer of this, some tiny little insects live on tiny galls. This gall that you see is about 1 millimetre across. The insect that's on it, and if we could make this movie work, is about 100 microns as an insect. I've shown this to entomologists and they just say, what's that? It's unknown. There it is, it's moving. See what a big beast he is. We went and re-investigated that tree to see if we could find more about this insect, and within about 2 weeks it had grown and gone through different life cycles, and now it looks quite different. And this one likewise is still alive. Most of the insects we've found, these galls are so small that to get the insect out you almost invariably damage them. There he is in all of his glory, and again no entomologists have seen anything like it. When we look at the leaf tissue analysis of Araucarias, we find that mostly they have reasonably good levels of nitrogen. Almost invariably they have very low levels of phosphorus and potassium. Cations, calcium and magnesium seem to be okay. Sodium is higher. These ideal ranges that we have come from the CSIRO data based on plantation Araucarias in Queensland. Likewise, we tend to find, now I should explain that these were in the Stirling area, these were in a park where 2 of the trees were very close to death, 2 of the other trees looked relatively healthy, and this is 4 trees very close to each other. Those trees that lacked any vigour whatsoever were particularly deficient in manganese, copper and zinc. Now those trace elements tend to become immobilised in alkaline soils, and they're also responsible for the uptake of potassium, which is important in the osmoregulation of trees. If we look at the soil analysis, we find that this soil is very alkaline. Recall that Norfolk Island has got fairly neutral pH soils, rich in nutrients. We're dealing with soils that are, firstly, the electrical conductivity is very high. These trees are in salty soil, alkaline soil. They have very low levels of nitrogen, but quite good levels of phosphorus and potassium and calcium and magnesium. Unfortunately, they've also got very high levels of sodium. When we interpret soil and the leaf tissue, we see that we've got a tree that's outside of its natural environment, not well adapted to alkaline soils, as many of our coastal flora is. The soils under the worst trees have got very high sodium, and this seems to be carried into the leaf tissue. They've invariably got very low potassium, and potassium is quite important for the osmoregulation, dealing with nutrient stress and dealing with water stress. The other, so in short though, there's no clear obvious connection, there's just a pattern that emerges. I've shown you just 4 of some 25 odd trees that we've looked at from good and poor environments. That pattern after 25 trees still doesn't become abundantly clear, but the trend emerges that we're dealing with a tree that's in an alkaline soil is in the worst condition. I've looked at the trees in good condition and they tend to be in much more acidic soils. They tend to be irrigated. Because we seem to see an association of Neophysococcum with the more rapid decline of trees, we are looking at treatment trials. Neophysococcum belongs to the Biosphere, which are abundant in the horticultural industry as pathogens of many important food crops. So there's many available fungicides that are known to treat them. Unfortunately, none of them are registered through the APVMA, so we can't use those without what we've obtained is a special permit for doing experimental treatments. Treating Araucarias with fungicides is not so easy because it is known from experience in both Western Australia and Sydney that these trees are very waxy. You can't put an aqueous chemical onto them because it'll just roll off. Ordinarily, you then add a wetting agent, a detergent, to make that stick. But Araucarias are very sensitive to detergents. People have used firefighting foam and there's been ocean outfalls that have killed Araucarias. So we were looking for fungicides that are systemic, that we can inject them into the tree and carry them up to the top of the tree. None of those are registered in Australia, but we do know that in the United States, a similar condition in spruce trees has used tebuconazole and propiconazole. These are triazole fungicides that are systemic and are specific for the Biosphere. The first trees that we injected really had been given a death sentence by arborists. We didn't want to kill trees with our trials. So these, the first trials were not successful in that the trees didn't recover. It was successful in that we could see no signs of phytotoxicity at the injection site. So that gave us the courage to go to the next step and look at trees that are in a less advanced stage. We injected 4 trees in the city of Stirling, going from almost dead to not very far off it to in severe decline to one that was just showing the first symptoms. The 3 that were in bad decline kept declining and have died. The one that was showing the first symptoms returned to good vigour. So we do have hope that fungicides may well be an important way that we can manage this problem. We've also recently tried a different type of fungicide, the strobin synthesis inhibitors. Azoxystrobin is one that is again not registered for these trees, but tends to have a broader range of fungal pathogens. It's now a bit too early to judge the outcome, but so far there's not really much improvement. What we're looking at now is a combination of both triazole and strobin fungicides with trace elements, because we know that trace element deficiencies are a major problem based on the leaf tissue analysis. Looking towards coordinating a management plan where we consider all of the factors that are there. I think one of the most important factors has been the climate change that Perth is experiencing. November 2019 was the hottest November on record. It just eclipsed November 2018, the hottest on record before that. We are getting a much longer summer, a much hotter summer. We've just come through the lowest rainfall winter on record. So I'm not exactly a climate skeptic. We're seeing climate change, and a tree that's already outside its comfortable range is now being pushed even further. I believe that that has a big bearing on the recent decline of Araucarias. Why do some trees do better than others? The more recently updated thing of this showed what we call, we do a phylogenetic fingerprinting on trees called a space analysis, single primer amplification of conserved elements. It is a genetic fingerprint of trees, or of anything that's multicellular, that shows how closely related to each other they are. Certainly the heterophyllars are closely related to columnaris, and that's actually not very different from bidwilli and Cunninghamii. But all of the trees in Perth are very closely related to each other. I don't think we've had a lot of importing of new genetic material. So there's not enough genetic difference to explain the susceptibility difference to decline in the Perth area. We're very sensitive about the idea of using fungicides to solve this problem because almost all trees have a native endophyte population. They're colonised by a microbiome that's in a symbiotic association with the plant, and most frequently they're staving off opportunistic pathogens and actually creating benefits. You know, not all germs, not all fungi are bad. So we're now using a technique called Oxford nanopore sequencing to characterise what the natural endophyte microbiome is. And thus far we've found that the trunk tissue, the cambium tissue in the trunk, is quite different from the branchlets, is quite different from the leaves. There is certainly an overlap, but fortunately with modern next generation sequencing, we can do these relatively quickly. Our nanopore system generates about 20 gigabytes of sequence data overnight. That takes about 5 weeks to actually make some sense of. I think given that many of the trees in the Perth coastal area are the responsibility of local government, then the development of a practical management plan that will address all of the different problems. Do they need trace element nutrition? Do they need watering in really hot summers? Do we want to keep these trees? I've spoken, because we deal with a lot of arborists, I realise that the actual removal of a tree is a very expensive exercise. These things are massive and they're in urban areas. They need cranes, they need lollipops, and they need people to pull them down very slowly. Increasingly, local government is also valuing trees based on its amenity value, its ecosystem function value. And some of the larger trees are worth upwards of $100,000. So the cost of removal versus the value of the tree is potentially, I think, we've in the news quite recently, I think it was the ABC 2 nights ago, showed tree after tree after tree being removed. Ratepayers being very upset at this. They're iconic trees, they've grown up with them. So there is an argument there needs to be a coordinated and a concerted effort to develop a management plan that's going to save these trees. That's it. Thank you.
Thank you very much, Peter. So we are now into lunchtime. So I'm going to take one question for each presenter and then provide you with email addresses if you want to get in touch with them directly. So do we have a question for Peter?
Thanks, Ben. Peter, we just wonder how much has been looked into as a fact of the primary stress on these Norfolk Islands being the salt freshwater interface. I know, for example, 15 years ago, got us way down there, the golf course in summer, that saltwater interface was going in past the golf course, the residential draw. You know, those bores are super shallow as well within the root zone of those plants. So I just wonder how that's been looked at as a factor.
We haven't looked at it as a factor in Araucarias because we're looking at trees that are very close to the coast and have been in that saltwater interface all their life. You're certainly right that the superficial aquifers in Perth are changing. We get an enormous amount of influence in that in our horticulture business, where because the rainfall has declined, aquifer recharge has declined. The nature of the superficial aquifer is that the water tends to be fresher near the surface. It becomes more saline the deeper you go down. There's another impact on the limestone aquifers that the water becomes more alkaline as well. And quite often there's a very fractal pattern of how this groundwater is being disturbed. Mostly the Norfolk Island pines aren't being irrigated and we have less rainfall. As I said, the ones that are on a hard surface, they've got roads nearby them, their roots must really struggle for getting water when the only thing that a tree can really use to overcome very high temperatures and low humidities is transpiring more water. If the water's not there, they're going to get under stress. So yes, water is a part to do with it, but that is quite complicated to actually figure it into the whole equation of a multifactorial problem.
Okay.